Go ahead, Miki.
Okay. Hello, everyone. My name is Miki Ii. I'm an assistant professor of biological sciences and neuroscience faculty at Delaware State University. So today I'm going to talk about our new project. And this project is basically to write an R15 grant, which this grant writing and mentoring opportunity is given by the MRDA grant I just received. I really appreciate the support from the Delaware CTL Accel program. So let me begin. So I'm talking about drug repurposing using C. elegans neurodegenerative disease models. Could you go to the next slide? Thank you. So in my lab, we have been working on the role of DNA repair factors and ribonucleases in neurogenesis and neurodegeneration in C. elegans. And we just began to work on translational research to contribute to the human health and to find new treatments for motor neuron disease, as well as frontotemporal dementia by using drug repurposing in C. elegans disease models. Please go to the next slide. Thank you. So what is drug repurposing? There are so many drugs for different diseases, but by using those already existing drugs approved for different indication, we can reuse by using for the treatment of another disease. And this is just an example. And this is basically showing the very different types of neurodegenerative diseases. And drug repurposing is helping the patients, different neurodegenerative disease patients, have their symptoms improved by using different types of drugs. Okay, so please go to the next slide. And C. elegans is a multicellular model organism which is widely used to study human diseases, very convenient, very simple, small, tiny little creature, which is a nematode. But it is good to use C. elegans because they are so tiny and economical and very well conserved genome. 83% of C. elegans genes are conserved in humans. Please go to the next slide. Thank you. So C. elegans researchers received Nobel Prizes. The highest level of research is available by using C. elegans. So 6 C. elegans researchers, especially very important findings are about apoptosis and RNA interference and GFP, green fluorescence proteins, to visualize neurons. All these are highest level of science. And in my research lab, we are working on apoptosis to find out how DNA repair factors and apoptosis pathway work together. And also RNAi for gene knockdown and GFP labeled neurons are observed under microscopes. Okay, so please go to the next slide. And the disease, especially my lab, is focused on motor neuron disease. One of the motor neuron disease, Icardi-Gutierrez syndrome, AGS. This is a similar type of disease. ALS is a well-known motor neuron disease, and AGS is a different types of neurodegenerative disease caused by the mutation of RNSH2 gene. And this is what we are studying. Please go to the next slide. And CRISPR genome editing is a very important area to make a precise change in the genome. And C. elegans was the first multicellular organism to use CRISPR to change the small changes in the genome. And we are using this technology to generate very the disease model in C. elegans. Please go to the next slide. And we were able to generate motor neuron disease C. elegans model strain, and it is the same mutation that AGS patients have. And we found that just one amino acid change of RNSH gene, that protein enzyme, makes a huge difference, like locomotion and pharyngeal pumping, which is food intake, as well as memory and learning. So this protein, RNSH2, is so important to control motor neurons and or movement of the C. elegans, and as well as memories. Okay, and also this mutant shows hypertrophy. A protruding vulva is a phenotype in C. elegans that mimics cancer development, a sign of cancer development. So both neurodegeneration and cancer, like pre-cancer symptoms, appear in these model worms. Please go to the next slide. And we began to work on drug repurposing by especially using anti-cancer drugs, because anti-cancer drugs have been used to treat Alzheimer's disease and other neurodegenerative diseases. And we selected several anti-cancer drugs, especially EGFR inhibitors and MEK inhibitors, and tyrosine kinase inhibitors. And the good thing about C. elegans is this is a whole animal model, and it's being used at pharmaceutical companies for drug screening. Okay, please go to the next slide. Thank you. And we got preliminary data recently, and very interesting finding is included in this slide. So I'm not showing many slides data, just one slide, but we found that some anti-cancer drugs does improve the symptoms of this AGS, neurodegenerative disease model worms symptoms, like hyperactive neurons, as well as hypertrophy, protruding vulva, are improved. So anti-cancer drugs do work to improve neurodegeneration in young adult worms and old adult worms as well. Please go to the next slide. So future direction is we want to accelerate our translational research using C. elegans disease models by focusing on high throughput screening of anti-cancer drugs and other type of drugs developed for other human diseases, and RNAi for gene knockdown. So this is mimicking antisense RNA therapy in humans. Also, personalized medicine is very important area. So we want to generate more disease model using C. elegans by CRISPR. And I'm going to write an R15 grant with the support of MRDA grant. Thank you so much. And thank you for your attention.